Topic today is the discovery of cisplatin. And the reason I wanted to talk about this is because I think it's an excellent example of how science works. I've always been intrigued by science ever since high school and college. And I asked the question, what is science? What's the scientific process? How do scientists discover those facts, those features that we use in progress of mankind? There are courses that go back at least 2 centuries in the philosophy of science, trying to figure out what scientists do, how they think, how this all occurs. And I've always been intrigued by the process itself. I know there's such a thing as the scientific method. We all know about that. But I wanted to dig deeper into what really goes on in science. So I think what I'm going to talk about today is a very good example that's put together in a nice package that gives at least one example of the way science works. Um, the um, the area in which here we go. Yeah, the area in which my topic occurs is in the field of cancer. Now, I'm pretty sure that most of you know what cancer is, or at least have some familiarity with what cancer is. You may have it, or you may have had it. Somebody in your family may have had it or has it. Any higher organism, or not any, but most, if not all, higher organisms, that is, organisms that have more than one tissue type in them, can have cancer. Cats, dogs, obviously humans, plants, particularly woody plants, lobsters, starfish, anybody can get cancer. So, what is cancer? Well, it has a number of hallmarks, but one of the main hallmarks of cancer is the fact that you have a cell that it usually carries out a process that is useful to the host organism. And this cell, for some reason, decides to de-differentiate or become somewhat more primitive in the sense that it now marshals all of its resources to divide. So that's one of the main things that pathologists look for when they look at any sample from a person, to see whether there are cells in there that are undergoing division more often than usually would be fine for that tissue type from at which the sample was taken. I've listed here the 4 main areas of therapies that have been used in trying to treat cancer in humans in sort of a historical order. The first therapy was using surgery, in other words, surgical therapy. And what happens there is that a surgeon goes in and cuts out a cancer and tries to eradicate the cancer by removing it from the host organism. In other words, a human, let's call it, we'll call it humans from now on. Um, so they're using surgical tools, scalpels, and so forth. They also can use lasers and other devices to try to kill the cells, that is, to burn or to kill the cells that are there. And this goes back at least a century, century and a half. The next therapy or therapeutic use was using chemicals or chemotherapy. And the first chemicals that were used came from plants and lower organisms, that is, bacteria, protozoa, so forth. Eventually, in the 1900s, chemical companies started to get involved in trying to synthesize chemicals that could be useful to treat cancer. Because remember, in surgery, you can only go in and cut out things that you can see. In other words, masses that are there, you can visualize. But cancer also can occur in the blood or in the lymph or in the interstitial spaces between cells, and so you can't get to that with surgery. So you have to use something else, and so chemicals were some of the was one of the ways that people try to go into to eradicate cancer. The topic that I'm going to talk about today comes from this area of chemical or chemotherapy. The next area of therapy was in using radiation, or what's called radiotherapy. When X-rays were discovered in the late 1800s, it was early on found that X-rays could cause the skin to burn. So X-rays were one of the first modes of therapy to try to kill cancer. And today, radiation therapy using X-rays, gamma rays, proton beams, meson beams, and so forth are used. Radioactive chemicals are used for the radiation effects. In other words, they try to target them to where the cancer cell would be and use that to kill the cancer. But most of the radioactive materials are used for diagnostic purposes. In other words, they send them in and then they try to image where the cancer is in the individual by looking at the signal that it gives off, the radioactive signal that it gives out. The final area is using immunotherapy or immunology. Now, this is the hot topic today. This started in the 1980s. And it grew slowly, but in the last 10 or 15 years, it has been the hot topic in cancer therapy. And there are a lot of developments that go on, I would say, daily in this arena. It's really a major topic. The field of oncology in medicine really got started in the 1970s as a separate medical discipline. And oncologists use all four forms of therapy today. So if one goes, if somebody has cancer, many times the oncologist will put up a set of protocols that will use one or more of these therapies today. In particular, for chemical therapy, you'll often find that more than one chemical is given. It's called a chemical cocktail. So it's not uncommon that people will get more than one chemical if that's what's deemed to be appropriate for the person. The discovery that I'm going to talk about today took place at Michigan State University, and it took place in the 1960s and early 1970s. In the 1970s, Michigan State decided that they wanted to establish a biophysics department. There were none or very few departments per se in the United States at that time. So they wanted to be a forerunner in forming such a department because they thought that this was going to be an important discipline in the scientific arena. They hired 3 professors. They gave them full professorships from outside the university to set up this department. The department was housed in what was called the Biology Research Building, which is a pretty small building, 2 stories high with a basement. Its main purpose was to house instruments that scientists from other departments could use, such as the electron microscope and nuclear magnetic resonance machine. But the department was put in there when they started. At the same time, Michigan State was building a brand new chemistry building to replace an old, outmoded building that was starting to fall apart. And the chemistry department was beginning to really expand because of the research being done in chemistry. So they were to build this building, and part of it was going to be devoted to the biophysics department. That is, the chemistry building was going to be too big when it was completed. So at least initially, the biophysics department was to be given a portion of one wing. So the department moved there in probably 1963, 1964, and occupied 3 floors and the basement of one wing. I joined that department in 1965 as a graduate student, and the department had at that time 5 faculty members, probably 2 or 3 postdocs, and approximately 10 graduate students. All the graduate students came from physics departments, except there was one that came from a biology department. We all pretty much had master's degrees. The purpose of the department was to train PhDs, but one of the main purposes from the university's point of view was to bring in research money, research grants. So research was the main focus of that department. The department offered 2 sets of courses, 1 year of lectures, 1 year of labs. And then the rest of the courses were things that were set up for each of the students in other departments. So we were taking advanced or early undergraduate courses in a variety of other science departments. let's see. Now, the reason I'm talking in some detail here is for two reasons. One is that some of the facts I give you will become important in this discovery that comes along. The second is to let you know that I was there when the discovery was being done. I wasn't in the lab where the discovery took place, but I was talking to the graduate students, the faculty members, the postdocs, and so forth. And we got together. We were somewhat isolated, so we got together socially a lot. We knew each other's families. We talked to each other daily on an informal basis. And so I wanted to give you a flavor that, I have a sense of what was happening, when it was happening, and hopefully this will come through as the talk goes on. Okay, now here's a diagram or a depiction of something, and anybody who's listened to some of my early talks knows that I try to get the participants involved a little bit in the talk. I want you to give me your idea of what this diagram is to depict. What do you think it is? Anybody?
Those look like the telomeres involved in cell division.
Yes. Okay. Good. That's one. That's one. I'll come back to that in a minute. But is there something else this fine?
A state transition.
What's that?
Like dipole flow.
Okay.
Two magnets. The magnet force between them.
Yes. Boy, you guys are good. You got it. Yeah. Without going too far afield, you got the two areas that I was hoping to find. Yeah. This is a diagram. You can think of it as being two opposite charges, electric charges, and what you're seeing is the force field between those electric charges. Or you can be looking at two poles of a magnet. So it's a diamagnet. So you're seeing the force field, the magnetic force field between the two magnetic poles. That's one way you can look at it. Physicists have seen this, in high school. It also can be is a diagram of what's called the mitotic spindle. When cells divide, this spindle forms. Now, picture a cell. If you follow my cursor, picture a cell around here. Okay. When the cells, we didn't want that right now. Hang on. Okay. When the cells divide, the genetic material will reside somewhat in the center of the cell, and it organizes itself. The DNA separates into its separate chromosomal features, and it also begins to unravel, and the genetic material then duplicates itself. As this process continues, finally, the duplicate sets of genetic material form into chromosomes. That is, proteins begin to come in and form together, and they form these chromosomes, which then are aligned along the equatorial plane of the cell. When that happens, this mitotic spindle forms, and the spindle is formed from contractile and structural proteins. And the chromosomes then move toward these cores. Okay, so once that goes here, once that goes there. At the same time, the cell membrane begins to invaginate or to pinch off toward the center. And by the time the chromosomes reach their cores, the cell has divided by the membranes coalescing here. So you get two cells. And then here's a new nucleus. There's another nucleus, and those nuclei then can divide by mitotic division. There was a book published in 1944 written by Erwin Schrodinger. This is the same Erwin Schrodinger who many of us have beat our heads against trying to solve the equation, Schrodinger's equation. He wrote a book called What is Life, based on a series of lectures that he gave at Dublin University in 1943. This book is publicly available today in HTML and PDF formats if you want to read it. And I fully recommend that anybody who's interested in the history of science to go ahead and look at it. I picked it up in between my sophomore and junior year in college. It's a paperback, and it's just as I was going through bookstores, trying to figure out what I wanted to read and so forth, I happened to see it, picked it up. I'd already had biology. I knew what mitosis was. I'd heard of Schrodinger. I'd never solved equations by that time, but later on I would do it. And I wondered what was in this book. When I started to go through it, I was mystified by some of the things that he was saying. In the past 10 or 15 years, I've picked the book up. I still have it. I picked the book up and read it again with the knowledge that I now have acquired in biology. And it's a very interesting book. A lot of the assertions, a lot of the statements that he makes in here, a lot of speculations are, if not incorrect, they lead in the wrong direction. But it's interesting in the way he's thinking. It's a fascinating book from that point of view. He did write a later book called Mind and Matter. I haven't read that one, but by the title, I think you can get some sense of what he's talking about with that one. He wrote this this was published in 1944. Physicists, of course, were enamored by Schrodinger. So I'm sure that a lot of physicists picked this up, read it, and were intrigued by what he was saying here. And my knowledge of history of science tells me that a lot of physicists begin to go to biology, probably somewhat based on having looked at this, saying, yeah, physicists can do something. It's an interesting area to investigate biology. Maybe we should do it. So you see a lot of physicists that went to biological sciences in the 50s and 60s particularly. Let's see. Yeah, like I say, pick it up if you have a chance, because it's a very interesting book. Now, the people who took part in what I call the basic science or the basic discovery are pictured here. First one is Barney Rosenberg. He's on the left picture. Of course, he's the male. He got his PhD in physics at NYU. He studied under Coleman, who I don't know anything about. It's Kallmann. Coleman had supposedly studied under Einstein. And I know that Barney really revered Einstein. Many times I've talked to him and this has come up. He was a solid-state physicist. Solid-state physics was really big in the 1960s. So he did electron paramagnetic resonance studies and so forth. And when he came to Michigan State as a beginning professor in biophysics, he started to do some experiments, EPR on biological molecules. The person to his right, our left, is Loretta Van Camp. She's a medical technologist. She'd gotten her BS at Michigan State. So she knew how to conduct biological experiments. She could work with bacteria, single cell organisms, as well as animals. So anything had to do with biology, she was very adept at. She is, I would call, she's very pleasant, very kind person. She was somewhat reserved, particularly around Barney. Well, and I'll tell you why in a minute. But she wouldn't take any guff from anybody, including him. But she was good, and she's an important character here. The third person is Thomas Krieges. He's on the right. Now, the picture on the left was taken around 1969, 1970. The picture on the right was taken 3 years ago. So you have to picture Tom like 50 years younger than you see him on the right, with a little bit more hair. He was a graduate student in chemistry. At the time of the discovery, he was trying to get his PhD in inorganic chemistry. His major professor wanted him, but didn't have any money. Barney had money, and he needed, as we'll see, he needed a chemist, somebody really that knew inorganic chemistry. So he hired Tom as a technician. So Tom was a technician working in Barney's laboratory in inorganic chemistry. Now, getting back to Barney, trying to describe it. It's difficult. I've tried for the last several days now trying to adequately describe Barney. Barney was a very forceful person. If he had an idea, he was forceful about promoting that idea. However, when it came to interactions with his students, postdocs, Loretta, whoever was in the lab, he was not that forceful. I mean, or pushy. He didn't micromanage. There were things that he would like to see done, and so they would have weekly meetings or monthly meetings and so forth to try to figure out where people were at, and that was it. He didn't micromanage. He didn't push people, and that allowed the lab to flourish and do things and go off in different directions that they probably wouldn't have if he had directed every single thing. And as we'll see, I think that the discovery would not have been made if he had micromanaged. Now, we'll get to the basic experiment that was done. On the left is a picture that was taken of the lab apparatus that was used. On the right, I'll talk about that particular piece of apparatus, which is actually incorporated in the one on the left. You can tell the one on the left has been squished in a lateral dimension, so it's that's not a perfect depiction. The basic experiment that Barney proposed to do was he had seen the diagram that I showed you earlier here, and he saw the similarities between mitosis and the electric and or magnetic fields. And he thought that mitosis perhaps had a magnetic component to it. That is, that during mitosis, there was a magnetic field that was established, and somehow mitosis, the movement of the chromosomes and so forth, were affected by this magnetic field. So he proposed to do an experiment where he would impress on that mitotic feature an electric field. He thought he might be able to alter the magnetic field with the inducing electric field. So he set up this piece of apparatus. Now I want to point out some features of the apparatus. First of all, if you look here, you see a signal generator, which is a acoustic or sinusoidal signal generator. So you can adjust the frequency at which you apply the signal. And so you're going to have a sinusoidal signal. It goes through an amplifier, and those of you who worked in this field many years ago will notice all the vacuum tubes here. Solid state, wasn't around then, so these are vacuum tubes here. So eat your heart out to see the vacuum tubes. They send the signal through a Tektronix dual beam, dual trace scope to the apparatus. Now the apparatus sits over here in this area here. It sits in this water bath. The water bath is at 37 degrees Celsius, or our body temperature, because the organism that was being chosen was E. coli B bacteria. It's a bacterium. In this chamber is immersed this device. And in here, they had media, which was put in to keep the bacteria growing. In other words, nutrients or things that they needed. At the same time, they would put in air. And so here's the air tank, compressed air tank. So air was put in, bubbled through the media to keep oxygen, nitrogen, so forth in the media. And as media was put in, of course, you had to have some way for the excess media flow out. So we have a flow out here. Okay. Now, to put in the electric, as he called it, field, he used platinum electrodes in here because platinum was considered to be an inert material to form electrodes. It was not copper or steel or tin or nickel or any of those that could perhaps react with the fluid, with the media that was there. So he put in 2 electrodes that were made of platinum because it was inert. carried out the experiment. Now, there's a couple things about this. One is he's using a time-varying electric field. He's not using a straightforward electric field. It's time-varying. So what is a time-varying electric field? Well, from Maxwell's equations, as I recall, time-varying electric fields give you a magnetic field. So really, he's impressing upon his cells a magnetic field. Sure, it's electric field this time, but it's time-varying. So he's got a magnetic field in here. Secondly, he's using E. coli. E. coli do not undergo mitosis. When E. coli divide, there's a certain, they're usually, the DNA is often circular. And so at a certain point on the circular DNA, they begin to unwind and the duplicate strands are formed, and this progresses around the circle and comes back to the beginning. And the two sets of genetic material then moved in opposite directions, and the cells divide. And bacteria usually have cell walls. They have some membranes, but it's usually cell walls. And these walls will pinch off and form the duplicate, or the daughter cells, as they're called. So there were two features about this experiment that always intrigued me. And talking to his graduate students, they would come down to my lab and we discussed things, and they would shake their heads too about this. What was his thinking when he was doing this experiment? I do know that he was planning to do that this sort of experiments here was being done sort of as a foreigner, just to try out the equipment, because this equipment had been loaned to them and they wanted to get it all working and so forth. But they were going to do mammalian cells later on. So this was a test run, and he was aware that he had to go for mitosis. So he was going to do mammalian cells later on. So that's the basic experiment to begin with. Now, here's the result of the experiment. On the left is a picture of E. coli as they normally occur. So the starting material would look something like this. When he turned on the field, he and Loretta found that the cells would not divide. They would sit there and stay there like this. So he was losing cells. There were fewer and fewer cells as time went on. So after an hour, they turned off the field. When they came back 8 hours later, they found what is in the picture on the right. In other words, what happened in that 8 hours is that the cells underwent genetic division, but they did not divide. Filaments formed, and these filaments got to be up to 200 to 300 times the length of a normal E. coli bacterium. For some reason, they had formed filaments in this experiment. So, why did that happen? What's going on here? Let me come back so you get a better sense of the particulars of the actual experiment inside that device. You can see here that we've got the media that's in here. He's got half cylindrical platinum gauze electrodes. He has media coming in, and of course outflow as the media comes in, the bacteria are dividing and so forth. He puts air in through a fritted glass, so he's got oxygen, nitrogen flowing through. He has an AC system, 1,000 cycles per second at 2 amps that he puts on for a while. Now, the medium itself contains, and it's called a minimal essential media for bacteria, contains no protein. It contains some inorganic salts, and those salts are potassium, sodium, calcium, magnesium, ammonium, chloride, sulfate. Okay, now these two, I cribbed this, as you can tell. These two are incorrect. Something is incorrect about these. The first one on the left is probably HBO4. In other words, hydrogen phosphate. Hydrogen phosphate does have a negative 1 valence to it, and it is a very weak acid. Dihydrogen phosphate, the other one on the right. This one, dihydrogen phosphate, does have a 2- valence, and this is considered to be a weak acid. This is all that's in this medium. I want to just say that it was lucky that they didn't use protein, and I'll tell you why in a few minutes here. Okay, so what's going on? What caused that? This is where Tom Krios comes in, because there's a lot of chemistry that they're going to have to do to try to figure out what's happening. Okay, first of all, they looked at, well, what's causing the filaments to occur? Is it some known form of filamentous growth? Is there something that is known that causes this? Because some of these things were known. So they ruled out osmotic pressure as a causative agent, temperature being wrong. It was at 37 degrees. That's normal growth temperature. Any dyes or penicillin, which is known to cause filaments? No, none are there. Any UV light present? No. Any other adaptive mechanisms that bacteria would use to form filaments? None that they were aware of. Were there any mutational agents known at that time? No, none there. So it couldn't be that. What about the electric or magnetic field, as it were, in this situation? Could it have been that caused it? And what they found out was that no, it was not necessary. Today, we don't know. Somebody I'll talk about at the end of this talk is actually going back and looking to see what was the effect of the electric field, if any. Anyway, at the time Barney and Loretta are doing this, it was found pretty early on that the media alone, when subjected to the experimental conditions, in other words, without bacteria, just putting it into a bath, turning on the electronics, letting it go, and then you take that media that had been conditioned by those experimental conditions, add it to E. coli, you've got filaments forming. So there's something that was in that solution that caused the filaments. Okay, what is it? Is oxygen necessary? Yes. If you put nitrogen or helium through rather than air or oxygen, through using the experimental conditions, then added that to fresh media, you did not get filaments. You needed air and or oxygen. You could do oxygen by itself or air. Nitrogen wasn't necessary, but oxygen was, so air was. What about any oxidizing agent that's normally present in the media? So what they did was what's called a starch Ki test, which is a pretty universal test in chemistry to see if there's any oxidizing agent that's present. And they found none. If you took just the media by itself, it was negative. Whereas they were thinking perhaps some electrolysis could have been taking place with those electrodes being in that media. For some sort of electrolysis might have been taking place there, and perhaps that was what was happening. But in order for electrolysis, you need some sort of oxidizing aspect to occur. But it wasn't in the fresh media. What about any of the components alone that are subjected? So, for example, if you put oxygen through that media, can you get an oxidizing agent? And they didn't find any. They used, for example, they use chloride perchlorate, and so forth that they thought might be coming from chloride, or the chloride ion, and they didn't find anything. It wasn't there. So, what was it that was in the media? They went down to find out what's in the media for the filaments to grow. And there's one or two elements or features or materials that are in that media that caused it. The ammonium ion by itself and chloride, or chloride by itself, was necessary. If you took those away, you didn't get media that would, when added to E. coli, cause the filamentous growth. So chloride's going to become an important material. The ammonium ion, perhaps. So this is what Tom and Loretta did. This took obviously several months, if not years, to go through all this. So they came up with that. They said, well, we think then that probably that the platinum electrodes are being electrolyzed. when you have oxygen, the ammonium ion, and chloride present to form this substance. This is a known substance, diammonium platinum hexachloride. It could be that that's the causative agent. That's what's causing the filaments to form. Now, this is a chemical that could be bought off the shelf. And when they did that and they put it into the E. coli media, they got filamentous growth. So, aha, this is probably what's causing it. The electric field is not needed. All you need is that chemical. However, during this time, there was a paradox that happened. In the early experiments, the growth that occurred when the concentration of the platinum agent in the media was 8 parts per million. Those experiments took place in the biology research building. Later experiments took place in the chemistry building. Fresh stock solutions later on showed that you needed the platinum agent to be greater than or equal to 10 parts per million. In other words, the early experiments, you could get growth at 8 parts per million of this agent, and it had to be 10 parts per million or greater for later experiments. What's going on here? What's causing that? Loretta had noticed something, and she wrote this down in her lab notebook, I think. If not, then she just recalled it out of the air. But she came up with, well, when the stock solutions of this agent are made up fresh, they're pale yellow. And the early experiments were carried out with this material at the biology research building, in which the windows faced southwest. Later experiments, which are in the chemistry building, did not have the sun exposure. Those experiments, in fact, that picture that I showed you before, is taken in a lab that is inside. In other words, it doesn't have any windows facing outside. It's off the hall, so it's totally dark except for the lights that are on. And in those experiments, when this material is made up into a media, that's when they saw that it had to be 10 parts per million. So it had to be something that was aging. So what's going on here? She noticed that when the stock solutions are left on the lab bench over the weekend, that those solutions became colorless. They didn't become colorless in the chem building. What's happening? Well, us physicists have some idea what could be happening, and that is perhaps there's a photochemical change that's occurring. So this was a hypothesis that was generated pretty early on, and a new postdoc in Barney's lab, Eugene Grimley, carried out this experiment. And to me, this is a beautiful set of spectra. This is an absorption spectrum of the starting material, in other words, the diammonium platinum hexachloride, which is now being subjected to radiation at 365 nanometers. In other words, it's over in this area. And if you look at the absorption spectrum as a function of time, you find that you start off here with the red one, that's the beginning spectrum. As time goes on, the spectrum changes so that, let me see, I've got the numbers here. Yeah, within 5 minutes, the spectrum has changed to that one. So there's some material has this absorption band in it that is being changed to this material. You're losing this one. And you get, when you get to a conversion of one chemical to another, you can get an isospecific point, this is called, where that occurs. So you're getting one material changing to another. But as you leave on this, starting irradiation, the whole thing comes down finally within, I think this was like 45 minutes or so, to this. It stays here. This is the final. So you get this isospecific point to begin with, but eventually this all falls off and you get this. So you've got something going on here. And the way to interpret that is that you've got actually 3 substances that are being formed. So you've got a beginning. In other words, the diammonium platinum hexachloride gets converted to something else, and that also gets converted under the conditions of this irradiation to a third chemical. So that was the result of Eugene's experiment. He got his chemistry degree at the University of Iowa. All right, so this is the interpretation. You take the platinum hexachloride, which has a -2 valence state, in the presence of ammonia, you can get rid of one of these chlorides and replace it with an amine to get a negatively charged species. And if you irradiate with 365, you can get rid of another chloride, and you eventually come up with a neutrally charged species, diamine hexachloride. Now, this number is wrong. I cribbed this, obviously, and this is wrong. This should be nanometers, not millimeters. Just try to be correct on those. Okay, so what happened is they came up with that hypothesis. So there's ways in chemistry that where you can separate these. You can use an ionophoresis technique to separate those three materials into their own, into separate components, and you can add these then to the media to see if it caused filaments. It turns out when they did the experiment that this starting material was bactericidal. In other words, the bacteria died when this was present by itself. Only this was present by itself. It died. Filaments did not form. When this one was present, it didn't do either one. It was not bactericidal. It was not filamentous. So something's not up. Scratch your head. What's going on? Then you finally get to this one, the neutral species, and that one is potently filamentous. They get lots of filaments that grow well when this one is formed. Okay, so at this point, Barney and his cohorts submitted a paper to Journal of Biological Chemistry. The referee turned it down, and the reason he turned it down is because in chemistry, the chemicals can form numbers of isomeric species. In other words, the three-dimensional structure of a chemical differs from one chemical to another. And if you're in biology particularly, let alone chemistry, that three-dimensional structure is really important. It is necessary. You have to understand three-dimensional structures perfectly to understand molecular biology. At least that's my thought, and I stand by it. It's really important to know that and to have that figured out. So the referee rejected it and said, you gotta tell me which isomeric form it is. So they obviously had to come back and do some experiments. Now, by that time, Andy Thompson, who had gotten his chemistry at Oxford University, had come into the lab as a postdoc. And you can buy the two isomeric forms, starting materials, to form the ending material that had been submitted in that paper. Now, platinum can form a number of compounds, and platinum being a heavy metal forms chemicals using coordination chemistry. And it will happen that for the trans form of platinum, this is the platinum itself here in the center. These represent the chloride ions. These are the amines, okay, NH3, the amines. That's trans. If you start off with a cis form, the chlorides are next to each other. The amines are next to each other, as opposed to being opposed to each other. Okay, so this is trans, this is cis. The chemistry, if you're a chemist, or if you follow some of the things I said before, you can get platinum in what's called a 4 state. In other words, you can have valence attachment above and below this band here. This actually forms a tetrahedron, or a square. So this is pretty much square in one plane, but above and below the plane, if platinum is in the four species, you can add chlorides, let's say. So with the hexachloride, you would have a chloride up here and a chloride down here. Okay. Now these two, as I say, you can buy them off the shelf, and it was known how to do the synthesis. So the referee wanted to know which one of these was the one that was important for what they had submitted the paper for. So they ran the experiment, and obviously good scientists are going to test these to see which one is the important one. But not only that, since they have starting materials, why not test those too? So they did. Went to E. coli. Turns out that the trans form of the chloride form, that is the trans diamine tetrachloride, is not inhibitory to cell division. You don't get it does not inhibit the cell division. The cis form, this one, is active, and it forms filamentous cells. In other words, this probably is and was what they were seeing in their initial experiments to get the filaments. The transform of the starting material has no effect, but surprisingly, the cis form is very active, much more active than the tetrachloride. So the cis diamine dichloroplatinum 2 is the key chemical substance, and that is what was called cisplatin. Okay. Okay, now, Barney had in his mind, right from the beginning, the idea that if he was causing these bacteria to form filaments rather than to divide, that he had a cure for cancer. And he went around really promoting that idea early on. I can remember him coming to the classroom and talking about this to some extent. We had no evidence at the time, but he thought that he had a cure for cancer. So very early on, Loretta, being a medical technologist, took on the task of trying to test cis-diamine or cisplatin, let's call it that now, to try that out against cancer in animals. Now, the NCI has certain protocols for just testing chemicals, and all chemical companies use this now. Pharmaceutical companies use it. So she tried this out in certain tests and showed that it had some activity. And it's interesting compound, perhaps it can work. The next slide I'm going to show is a slide of some animal experiments. And if any of you are queasy about looking at animal experiments, I recommend that you close your eyes or something like that, and because I'll talk about it and I'll show you the results here. All right, this is an experiment that was done probably within the first few months. It took Balb C mice, and they inserted sarcoma 180, which was a known solid tumor that grows well in mice. And they have two sets of mice here. Let's take this one. This is the control animal, and you can see on day 8, after the sarcoma was implanted in the peritoneal cavity, you can see it's growing. By day 12, it grows more. By day 16, it's even more. By day 20, it's really getting big. All of the animals in this cohort die within 20 to 30 days. In the experiment that they did here, on day 8, they injected this animal, which you can see has the sarcoma in it, same as the control, with one injection of the cisplatin, 8 milligrams of cisplatin per kilogram of body weight. Single injection. On day 12, it begins to regress. On day 16, it's almost totally regressed. And on day 20, it's gone. By day 36, it looks like it could have come back again. But they got a lot of cures in this one. And this was the experiment that really got NCI excited, because this had never been seen before with any chemical. They'd never seen a cure of this type, cure so-called, of this type. So they became somewhat excited about it. Now, if you ever deal with government agencies and so forth, you know how tough it is to get them to move. It's just there. It's good, but it's there. You have to work with it. Barney, being a forceful person, really pushed the idea of cancer chemotherapy using cisplatin. And he went out and really tried to sell people on it. NCI became interested in it. So he would call NCI every month and ask them about, well, what's going on with my compound here? What do you know? Who's doing what? And how are things working out? At the same time, he went across the pond and he enlisted people in Great Britain and in Europe with his ideas and thought, tried to get them to do things. Sure enough, people in those various places heard about it, latched on and started to do experiments themselves. So there was a lot of work that was being done in the world, in the United States, MD Anderson in Texas, some people there were working in cancer. They started to do some of the work. In Great Britain, it was the Wadley Institute who did a lot. And during this time, Barney actually set up, along with his wife Rita, a yearly set of meetings where they would invite people from all over the world to come in and talk about using cisplatin as a cancer chemotherapeutic agent. And the meetings were robust. There was a lot of work being done at the time, trying to get it to try to find the efficacious concentration and the protocol for administering it to humans if they could do it, but particularly to animals. This went on for some time. There was a lot of testing that was done, and phase 1 trials for the cisplatin took place in 1972. Now, this discovery was done in like the mid 1960s or so forth. A patent was filed and was allowed in 1968. In 1972, as I said, phase 1 trials for the cisplatin took place. By 1974, they had worked their way up to phase 3, where you now start to give the chemical itself to individuals who may be treated with it. And usually these people have already been subjected to other treatments for their cancers. This is a picture of Larry Einhorn on the left and John Cleland on the right, taken 3 years ago. Larry Einhorn is an oncologist. He'd gotten his training at MD Anderson, and he was at the University of Indiana at the time of the critical result. John Cleland was like I say, this is 3 years ago this picture was taken. John Cleland was a 23-year-old man who had developed testicular cancer. He had undergone surgery where they removed one testicle, and it seemed to be under remission for a while. But testicular cancer is very insidious, particularly in young men, and it kills almost everybody, in any young males who come down with testicular cancer. And so after a few weeks, the growth came back in that area. So they started chemotherapy. And there were some agents that had been shown that had some efficacy, so they tried this. So Larry tried that and gave it to John. And sure enough, it caused some remission of the cancer. First one did, but then the cancer came back. Tried a second one, came back. Third one, it came back. And this is within months. This cancer really grows. By that time, John had also developed metastasis to the lungs. So there were some pictures of John's lungs that were really almost opaque. Now, Larry said, okay, we've got this new chemical. It's been tried once before on somebody, and that person died of kidney failure, but it was not due to the toxicity of the platinum compound. It's just the person was going to die anyway. Probably, I mean, subsequently, yeah, they found out that, you know, his kidneys had failed, and so he wasn't going to live anyway. So he said, we can try it with you because it's almost a last ditch thing. So John says, well, what have I got to lose? So he says, okay, let's do it. So Larry gave him the protocol for the cisplatin. And it turned out that 10 days after the injection, Larry's chest x-ray came back normal. It just blew their minds. It blew all the people in the area minds that had happened that quickly. Subsequently, it turned out that his testicular cancer was gone. And as far as I know, he's still alive today. That's an amazing story. Now, the FDA approved the cisplatin in 1978. Subsequently, there has been a lot of experiments done, a lot of work done, in taking platinum compounds and trying to find out better compounds that work, and B, in doing oncology, in other words, trying to figure out the protocols of giving these compounds to patients in which you get successful results and the patients live well. Platinum is a heavy metal, and that was one of the reasons why NCI did not want to work with platinum initially until this sarcoma 180 experiment came to their attention. It just thought that it was like mercury, lead, other things. It poisons and is not worth it, so we're not going to do anything with it. But since you had this effect, this curative effect, then they started to work with it. So platinum has been researched from both the medical standpoint and the synthetic chemistry standpoint for a number of years now. So today we have being used two chemicals with a platinum base, carboplatin and oxaliplatin. And I think there's a third and fourth one that may be on the market today. I'm not real sure. There's lots of them that are there. There's literally hundreds, if not thousands, of platinum compounds that have been synthesized and have been tested for cancer. A number of Barney's postdocs, none of his grad students, but his postdocs went on and they spent their entire careers studying the synthesis of platinum compounds that could be used in chemotherapy. And there was one person in Europe, in Germany, who was a physician, beginning physician, and was going to be actually an oncologist, who took it upon himself to try to get the platinum compound to work properly. And he spent his career in oncology, and he's now well respected. He's come up with an equation that's used often in oncology by studying the platinum compound. So this is a field that is sort of hidden from public view to a great extent, but it is big. And carboplatin was also developed at Michigan State by some postdocs and Barney. And in this, you have what's called a malano species here. These places here have carbons. You have a double bond oxygen, single bond oxygen. These are all carbons here. This is a three-dimensional view of that. It's hard to see, but you can see the amines over here on the right, and then you see the carbons in red. The oxygens in red, carbons in black, and hydrogens. If you don't see anything off here, those are hydrogens in order to complete the valent state of carbon. And you can see there's a torsion of this tetragonal material or box-like material. Has to rotate. Oxaliplatin, and what they've done here is they've looked at the nitrogen side of it, and they've made some changes out here. So you got a cyclohexane off this, and they've worked with this, and this is now being used. They've taken off this 4 carbon species on this side. So this is another one that's being used. So it's quite common now to see these compounds out there. It is so common that today this chemical compounds have been found to be effective against testicular, obviously is an important one, ovarian, cervical, breast, bladder, prostate, head and neck, esophageal, stomach, colorectal, lung, kidney, brain, neuroblastoma, bone, Hodgkin's and non-Hodgkin's lymphomas, mesothelioma, melanoma, multiple myeloma, and leukemia. It's used a lot. It's one of the major chemical compounds that's used. From what I have seen and heard, somewhere around 40 to 50% of chemotherapy uses a platinum compound in some form. One of the major problems with cisplatin, which was known at the beginning, was this toxicity. It does cause renal toxicity. It also causes toxicity of the hearing system. In other words, it goes to the brain. And so what oncologists, research oncologists do is they try to find a way to alleviate the toxicity. If they can find a compound that will cure cancer, or at least cause cancer to go into remission, they'll try to alleviate any toxicity they can find. You try to find a compound that works without toxicity, but if you can't, then you try to alleviate it. And so there's been a lot of resources that are put into that. And that's why these compounds are so prevalent out there, because they have done both of those. And these two are much better in terms of the toxicity is concerned. The carboplatin was given FDA approval in 1989. Now, carboplatin and cisplatin had patents issued for them that Michigan State University owns. And from the royalties of those two alone, Michigan State has made between $300 and $500 million over the course of the time since the 1970s when they started being used, which is a pretty healthy amount of money for this kind of a material. So Michigan State very, very proud of that, obviously. Okay, just as a side note, or sort of a coda to what I'm talking about here, there's two people I want to discuss here for a second. First is James or Jim Heschley. He got his degree from Michigan State in chemistry. Went off to Oak Ridge, some other places, excuse me, and then came back as a postdoc for Barney's lab. So he was in Barney's lab between 1960 and 1971. He's taken it upon himself to really pull together the information, most of which I told you today. And he's published a little monograph about that. And he's the one that I've talked to a fair amount. Because, like I say, I knew these postdocs. Our families knew each other and so forth. So Jim, I have to give him a lot of credit for a lot of things that I told you today because he has the lab notebooks, and I've discussed that a little bit with him, and so they're there. On the right is Tina Rosenberg. Tina is Barney's daughter. She went to Northwestern in what was it? In news, whatever that I suggest. But anyway, she is she's a researcher in like sociology, but she got a Pulitzer Prize in nonfiction in 1995 for a book called The Haunted Land: Facing Europe's Ghost After Communism. She went to Eastern Europe and interacted with the people there in the various countries: Hungary, Poland, Czechoslovakia, East Germany, and so forth. And she wrote this book about how they're coping with life after communism, after the Berlin Wall fell. She got a a Pulitzer Prize for that. She's right now on the editorial, she writes editorials for the New York Times. So occasionally you'll see her byline in the New York Times as having that sort of prestigious award. Okay, so at this time, I can close it now. Oh yeah, okay, one thing I wanted to do. I was talking about the scientific process. What is the scientific process? And so my conclusion, based on this story, as well as my observations of various physics, chemistry, biology labs, is the scientific process has these features to it. First of all, the person or the group have to have a real innate sense of inquisitiveness, have to be constantly questioning all the time. That's what a good scientist does, as far as I'm concerned. For the process to occur, there are a number of features to me. One is serendipity. There was a lot of those results that I just mentioned here that just, it was luck that they occurred. We would not have gotten to where we are today with this compound if it hadn't been for luck. Just happened to happen at the right place at the right time. You have to have tenacity. You have to go after it. You cannot be burdened by a negative experiment. You have to keep going. Somebody says no to you, which happened to Barney a lot. He just kept after it. Like I say, he was forceful. He had tenacity. You have to have logical thought processes in science. That's extremely crucial. You have to be able to think logically. You have to be able to carry things out and think from one step to another, logical processes, from your computer stuff, your if and statements, if then and so forth. That's really important in science. You have to have background knowledge. You have to be pretty well attuned to your field. So Tom Krigas being a chemist, he knew the inorganic chemistry stuff pretty well. He knew where to go to find out how to do those experiments. So you have to have good background knowledge. And finally, you have to have what I call keen observation skills. If Loretta hadn't made that observation of the source material changing color over the weekend, we would not probably have gotten to the cis compound, the dichlorodiamine compound, if it hadn't been for her observation there. So you have to have keen observation skills. And finally, because of all this happening, I think this is all enveloped in what I call crazy thinking. You can't be constrained by what is the mode of normal thought in out there in the gestalt. You have to be able to think beyond that. You got to have some crazy ideas, and you just keep going with that. So it's a very fascinating field to me. I'm happy to have spent my life in it. I think it's a fascinating thing, and I'm happy to have been a scientist and work with scientists.
You mentioned in your talk that thank God they didn't put a protein in the solution. What was that comment? What did you mean by that? What would have happened if they had protein in there?
Yeah. Platinum is, well, metals are reactive. Biological materials have a lot of different chemicals in them, obviously. And platinum reacts very avidly with sulfur that is in proteins. In other words, sulfur that is attached to two carbons. So if they'd had protein in there, that platinum would have reacted very avidly with the protein that has the sulfur in. The working hypothesis, which I didn't go into, is that platinum has an effect on DNA. And if you want to talk about that, we can go into that in a little bit more detail. So they think that's why division doesn't occur, both or particularly in the cancer field. They think that that's what's going on. That the DNA, the interaction with DNA, is what's causing the cancer to be cured. And we would never have gotten there if the proteins had been used in that media, because the platinum would just react with that and never done what it did otherwise.
Was the absence of protein a conscious decision, or it just happens to be the way the medium was configured, and that's how they use it?
Yeah, it's because of the way. Yeah, they weren't conscious of it. The media comes that way. I mean, that's what everybody. The media that's used for bacteria grow does not have protein in it. Contrary, when you go to growing mammalian cells, there is a lot of protein in that media. So if they'd started off with mammalian cells, they wouldn't have gotten the result that they saw. So it was unconscious. Again, serendipity. It just works out like that.
Yeah, great, thank you.
Dick,
Yeah.
what about other platinum group compounds like, iridium based, for example?
What based? I'm sorry.
Iridium, for example.
Oh, you mean using something other than platinum as the base metal?
Yeah, but in the same group.
Yeah, in the same chemical group and things like that. Yeah, that's been looked at, and the closest they've come is palladium, but platinum is by far the one that is the most reactive and has the best activity. All the others have not worked. Osmium has sometimes used, iridium. Like I say, palladium is the one that is the closest, but it has nowhere near the activity that platinum does. So again, this turns out to be platinum because he used a platinum electrode.
But do we understand how it works? Why what it actually does at the level of the cells?
Yeah, good question. We don't know for sure. Like I say, they think that it interacts with the nucleic acid, with DNA. But platinum, I think I told you sometime before, I was in Barney's lab as a postdoc because he took me on when Nixon cut off my NIH fellowship. And so he took me on. I established a cell culture lab there in his group. And with electron microscopists and we showed that the platinum gets into various organelles within the cells, including the nucleus. So it could be anything. Now, one of Barney's graduate students early on showed that platinum inhibits DNA repair. Your DNA is being repaired constantly by a number of processes, biochemical processes, constantly being repaired. It's constantly undergoing mutations and alterations, and it's constantly being monitored. The platinum compound early on was shown to inhibit one of the major repair processes. So we thought that platinum went to the DNA. A second graduate student, a very good friend of mine, showed that platinum reacts preferentially with guanine. Now, when I give my talk on CRISPR, I'll go into these bases a little bit more and show you what they are and what they're formed of and so forth. But guanine is one of the 4 bases of DNA, preferentially over any of the other 3 bases in DNA. So it's guanine. Subsequently, in 2000, I think 15 or 16, a chemist at MIT and his graduate students and postdocs who does X-ray crystallography showed that the platinum compound actually causes cross-links between two neighboring guanine, what we call residues or bases. And these guanines can be on opposite strands, in other words, across from each other, or, and I look like more preferentially, between 2 guanines that are next to each other on the same strand. That seemed to be the preferential area. So right now, that's where most people think that the platinum compound is having its action. However, platinum, we know, can be transported by a variety of actions within the cells, and the way things move around in cells is an active area of investigation today. So it's possible that the platinum compound is actually carried from extracellular, notice when it's injected, carried from the extracellular area through the cell cytoplasm to the nucleus and acts there. Or it could be acting in some other area prior to DNA synthesis or DNA repair and so forth. So that's an open area of investigation, but the present hypothesis is that it's acting directly with chemical reaction at DNA.